Probably going to have more than 4 tests.
Okay.
Because, these chapters are huge. And if we do 4 tests, we've got 15 chapters. And so you're looking at 3 to 4 chapters per test. If I did 4 tests on this one, you would cover, 120 pages in the book? Something like that. I mean, and the amount of information is just...
It'd still only be 50 questions, right?
It'd still only be 50. There only be 50 questions. On 4 of the tests, you're going to have 10 lab questions.
Okay, so how many more tests are you anticipating having? Because in our syllabus, we're supposed to have 4 and draw.
I know, but then what I will do is I will have to change the syllabus, and I'll have to give you that in writing. And that, hopefully, I'll have it by Thursday.
So how often?
But I think probably every 3 chapters, which means you'd have 5 tests.
Okay.
How are you going to do the point thing?
Well, the grade is actually set on a percentage. So if I have 5 tests, if I have 5 tests at 50 points a test, instead of the 100, and you can drop 1, instead of the 150 points you're going to need there will be 200 points. Now, there's pluses and minuses. You're still going to be able to drop a test. There's still going to be 50-point tests. But when you go to study for a test, if you do poorly, you've got an extra test to try to make it up on. If you're doing well, you're doing well. But the thing is, the ability to absorb 4 chapters worth of material and try to regurgitate it on a test is, that's pretty overwhelming.
But to be honest, that's kind of what I feel like right now, because we're getting tested on Thursday on 4, 5, and 6, and we're going into 8 today. And then we only have 7, 8, so we just have 9, so we're going to have another test right off the bat.
There will be,
I almost feel like this is harder because we have more tests.
Yeah,
personally.
It's just really
Because
tough.
we still have only 15 weeks to finish 20 chapters, whether we test it every week or once a month. We don't have any extra time to study chapters, just because it's chopped up.
I'd rather have it back to the 4 versus 1.
well, we're obviously not going to do 4 on Thursday unless you want me to throw 7 into the test on Thursday. So then we're going to have it with 5, some of them. 5 chapters.
Why is that? Because we should be all caught up.
We're actually ahead of Mary's class.
We are ahead of Mary's class, but Mary is way behind on the, I'm not testing in synchrony with Mary's. My tests aren't the same.
Oh, no, I just thought you were trying, because you always said we're going to get behind. I thought you were...
I'm trying to make sure we get through all the classes before the end of the semester, all the chapters before the end of the semester.
Okay, I just...
And, you tell me, if you want tests over 4 or 5 chapters, we'll go back to that. The next test will be probably 5 chapters. 7, 8, 9, 10, 11, and 12.
Right now, it's like every week we have to be studying hardcore for a test, whereas
Yeah, it's harder.
it's
Versus
sometimes
we have a
like
weekend
I
to
have
get
to
it
work
done.
everything.
Well, we have 15 weeks in the class. We have 20, it's a test every 3 weeks.
But if we change it, it's going to be almost a test every week.
Not if I go every 3 chapters.
So when will we have... Because we're already on we're starting chapter 8 tonight.
We're finishing 7, and we'll just get into chapter 8. We won't finish chapter 8 until next Tuesday, I expect, and maybe start on chapter 9. Chapter 8's a long chapter.
Chapter 7.
I don't know. For me, I just feel like I'm overwhelmed with the fact that we're studying for chapters that we talked about a long time ago, and we're moving on to chapter 8, and you're telling me how hard it's going to be, and then...
Well, chapter 8 is not hard. It's just a lot of memorization.
Okay, but that's hard to study for in such a short period of time when you're trying to cram. Because right now, I'm not even focused. I can tell you right now, I have not paid attention to chapter 7 because I'm having to focus on 4, 5, and 6.
That we're being tested on in today.
Like, so we take the test on 4, 5, and 6. Or, I'm sorry, 5, 6, and 7. And then after that point, will we be learning, 8, 9, and 10, and then have the exam? Because, I see where she's coming from, because we're learning about 8 and and 7, but we need.
Okay, the original intention was to have the test on 4, 5, 6, and 7.
Right, but that.
And-
Well, actually, wasn't it, we're supposed to have chapters 1, 2, 3, and 4.
Well, when Mary gave me the original test, it was only 1, 2, 3.
But you didn't take her test.
I'm not using her test, but I've seen her test.
But she tested on chapter 4.
I know that. I'm
I'm
not,
just confused.
yeah. Well, what Mary does, my class is independent of Mary's, okay?
Okay.
So, if you want only 4 tests, then you're going to have some tests that are going to cover 5 chapters.
But wouldn't we be caught up though? Because we're only
You've
behind-
got 4 tests and 20 chapters.
But we're only behind one chapter because that chapter 4 was supposed to be on the first test.
Okay, but
so
you
this
still
test would
have,
just be 5 chapters.
you still have 20 chapters and 4 tests. Plus the final.
We do a vote?
No.
No?
Well, that would only be 5 tests. Well, if you're counting the final as a test, yeah, then 5 tests.
So that's 4 tests per chapter.
That's, yeah,
4 chapters
yeah,
per
yeah.
test.
What it was set up as. I just am nervous that this system that we're in, like I said, I can't speak for everybody else, but I believe I'm not reading chapter 8, I'm not reading chapter 7 right now because I'm trying to remember what was on chapter 4 that we talked about a really long time ago, back 5, 6, you know what I'm saying? It's like, chapter 5 is kind of a complicated chapter. I mean, your review sheet helps, I appreciate that, getting that as a bonus.
Well, we should be caught up, right? After this week, once we take the test on Thursday, we should be caught up. The next test.
Yeah, you would have a next test fairly, 2 weeks you'd have another test.
7, 8, 9, right?
7, 8, 9.
And then we'll finish 9 before the next test.
Or 7, 8, 9, 10.
Okay.
But that's in 2 weeks. Is what you're
But
saying?
what-
Yeah, 2.
Okay,
What makes it
how soon after we finish a chapter are we?
Well, if I finish the chapter on a Tuesday or a Thursday, I'd like to have the test on the next week. Well, you're still relatively.
Because that's how we're doing it.
Yeah, right now, it's very,
Well,
very
that's
long.
because, when I asked you, we can put the test off until Tuesday and I can throw chapter 7 in. And then you got your 4 chapters. And we're finishing chapter 7 today. That puts us right on track with what you want. But you've got 120 pages of things to test on.
For me, my concern is not this test, it's the next test. That's why I'm worried about, personally.
Well, at some point, we got to catch up.
We still have to know all of it.
I just feel like it's not, I feel like we're being punished
Catch
for
up
the
to what,
mistake.
to who? Catch up to what, to who?
To catch up to a timetable that will get all the testing in and not load up on, like 5 chapters per test before the end of the semester. Now, if you're concerned about waiting too long from the end of the chapter till the test, we push this test off till Tuesday, and I'll test through chapter 7.
I think I understand why she was referring to Mary, just because if she's able to hold back, where is she cutting off later? She has to be cutting
Well,
off.
again, this class is not Mary's class.
No, but I'm wondering if someone, if the teacher is able, I'm just questioning the concept here that you want to get the 20 chapters in, but if she's able to hold back and wait for so long, where is she cutting? Is she?
I don't know.
Cutting chapters?
This isn't Mary's class.
She's not cutting off.
She's not going to cut chapters.
No, okay.
So,
Then they're gonna just struggle later on.
They're going to get, she's a couple of chapters behind us.
Sometimes she omits a chapter.
this is like how this original syllabus is set up. Our second test date was supposed to be 3/15, which not
Next week.
Thursday. And then test 4 is supposed to be April 12th, so it's about a month in between. Versus we're doing 2 weeks in between. That's what my problem
Not
is.
even 2 weeks.
The 2-week chapter difference on this test. Because here it's set up for almost a month. And that's a lot of time to absorb the information, go over it, read it, it's on a more comfortable schedule versus we're having the test on
Yeah,
8th, 9th, 10th.
it's set up for a month, but it's set up for more chapters and more testing.
But I don't feel like it's our fault that our first test didn't have chapter 4. Do you know what I mean? I'm not saying it's necessarily yours, but I feel like we're being punished for being behind and we didn't do it.
Actually, you did take the test.
As I said, the reason we're having this test is I didn't want to test you on 4 huge chapters.
I'm happy with
I mean,
that.
that's the bottom line there. You want to get tested on 4 chapters, I don't have a problem with that, but you're going to have a lot of work to do, as opposed to testing on 2 or 3 chapters while it's still fresh.
How many questions are you going to put on
There are
chapters?
50 questions per test.
So whether you're testing us on 4 chapters or 2 chapters, it's still going to be 50 questions?
Yeah.
Oh, then I'd rather go with the 4 chapters,
Yeah,
50
because
questions,
that's
plus
right,
questions
that's
per
more
discussion.
being tested on
There's more questions, exactly.
More detail.
That's more detail.
far more detailed.
Can we just stick the way it was and somehow make up that chapter 4 separately?
Let's just take it on Tuesday and do 4 through 10.
Yeah, I thought they were 50 questions.
If we get through 8 today, I'll do 4 through 8 on Tuesday. We get through 7 today. We know we're going to get through 7 today.
4 through 8, 5 chapters?
They're huge. We'll do 4 through 6. You want to do 4 through 7 on Tuesday?
Go back to schedule.
Well, I would rather just stick with the syllabus.
Because we're going to have 50 points on, instead of having a test of 50 over 4 chapters, we're having 2 tests with 50 points, so it's 100 points now.
So basically 10 questions per chapter versus 25 questions per chapter.
Yeah.
No, there's 3 chapters on this exam.
3 chapters, 15 points.
15 points.
The test actually has 50 points plus 5 extra credit plus 10 lab questions.
And the labs we haven't gotten back, so I remember
We're
we
getting labs back today. You'll get them back today or Thursday. Now, are we doing chapter 7 on Tuesday? Let me see a show of hands.
What, can you explain what that means?
Will it be 4, 5, 6?
4, 5, 6, and 7 on Tuesday, as opposed to 4, 5, and 6 on Thursday. 50 questions.
Versus having 50 questions on Thursday, and then in 2 weeks having 50 more questions on 3 more chapters.
Right.
I'd rather do Tuesday with 50 questions on paper 7.
50 questions on Tuesday.
And
Yeah.
What's the question?
50 questions through
Drop it.
4 through
You
7.
have your idea, don't pressure anybody.
The tests are 50 questions.
I'd rather go 4 through 7 on Tuesday like the syllabus.
Right.
Information.
No, it's not really very much. Did you write the test? Yeah, did you write it?
What's that?
Did you write the test?
I'll have to rewrite the test.
You did the test already.
Left questions for test that you guys voted. So.
Okay, hold it. Okay. The test will be Tuesday on 4, 5, 6, and 7. You got the weekend to study, but you're going to pick up an extra chapter. That'll put us online pretty much with the syllabus. And whatever we're done by the time we hit the 12th of April, you're going to have a test on everything since then.
You mean a cumulative test?
No.
8, 9, 10. If we get through chapter 12 by the time we hit the 12th of April, but the 12th, you see, you got a week off.
Yeah.
So if I turn around and give you a test on chapters that we cover by the 12th of April, it may be a 50-point test on 2 chapters.
Why would that be?
Because we take a test on the 15th of March, you have the 22nd, 29th, and then it's the 12th. You only got 2 weeks between tests.
Why don't we put 7 on that next one and then do the most then, because we have a week to study off of break. I mean,
Just give us study on break.
Yeah.
Just a quick question. If you added another test, doesn't that kind of mean like if it's more points, it's more of a buffer room for our grades especially?
Sure.
So wouldn't that be?
I mean, you've got a chance to make up, or
If he adds in another test, that means there's more points like total, so it's more of a buffer room if you messed up on something.
well, it's more of a buffer, and we're wasting a lot of time. It's more of a buffer, and you're being tested on on less chapters. Yeah, you're going to have to know the chapters, but you're going to know the chapters anyway.
So let's just do 4, 5, 6 Thursdays and 7, 8, 9, 10 on Wednesdays.
You want to do 4, 5, 6 on Thursday, 7 through 10 on the 12th of April.
yes.
Yeah.
Yeah.
Okay.
If that works for you.
7 through 12?
10.
7 through 10, unless we get beyond 10.
We're just adding the at the end instead of, yeah.
You're going to add the 7 at the end on the next test.
And then we'll kind of be on schedule.
No, we still are going to have a couple of 5-chapter tests.
Well, yeah, we do that after the 12th, right?
We do the 12th, you'll have 7 through 10, or unless I get beyond 10.
Well, tonight, could we just finish 8 or 7 and then do like a full good like review?
Tonight we'll finish 7, we'll start 8, and we'll take an hour for review.
I'm confused what we're doing. Do we have a test on Tuesday or Thursday?
You have a test
Thursday.
on Thursday,
4, 5, and 6.
4, 5, and 6.
And then April 12th.
April 12th, you will have a test on 7, 8,
And what?
9, and 10, hopefully. On the 12th, it's going to be 4 chapters if I'm through them, yeah.
So then
We're almost through 7, so we'll have 8, 9, and 10.
It will be much better if we take it for Tuesday because it's still fresh. The chapter 7 is really long.
Chapter 7 is very long. So let's do 4, 5, and 6 on Thursday.
Well, you have to decide as a
And
class
that's
because
7 on Tuesday.
So instead of a test every 2 weeks, we'll go to one every 5 days.
Every other, 5:25.
what about if you say make write down the options and after break we come with a conclusion.
I'll see why
Why
we
don't you
can
just
just
make-
You know, I'm sorry, but time out.
You're gonna need to make a decision because
We will have 4, 5, and 6 on Thursday. On the 12th, we will have 7. On the 12th of April, we'll have 7 till through whatever we've covered by the Tuesday before that.
And then can we just have like a really big review for that? Like maybe like go over 7 again? Like,
Well, we'll
I mean, there's
have,
going to be 50
A big
questions,
review
right?
for
So I mean,
3 chapters?
like,
what?
A big review
Are we
for
worried
3
about
chapters? Is that what, because we're just having 3 chapters, right? 4, 5, and 6?
We'll have an hour review tonight. On the 31st, after lab, we will try to have another hour review. But then you've got 10 days to study for the test. And we will test up to whatever I cover on the 29th.
Yeah, but
Okay.
you know what's so on the, you know how you said April? If you do 7 and 10, that sounds fair because 7 is really large. But then you're saying if you go farther than 10,
We
you're
have a
going to
week
do.
off.
But 7's large. That's
I know,
going to be hard.
Okay, that's what it is. That's just what it is. We got to catch up
Are we going to have
sometime.
the dates of the exams, like,
I'm going to get it back to what it is on the syllabus.
okay.
The chapters may not match, but the exam dates I will try to match
That
to the
makes it easier
syllabus.
when we know the exam.
and plus there are only 50 questions, so I mean it's not going to be that many questions from chapter 7. You know?
but you have to study them anyways. For me, there's no.
No matter how you break it up, we have 15 weeks to do
Stop.
20 chapters, period.
Stop. We got 20 chapters. We got 15 chapters. 20 chapters.
20 chapters. Right.
And we have 15 weeks.
No matter how we slice
No
it.
matter how you slice it. You're going to get tested on all the chapters, one point or another. You can have a big test and have a lot of chapters on the test, or you can have smaller tests and have fewer chapters. But right now, we're going on chapter 7. Okay. Let's go back a little bit. We talked about neurotransmitters. Your action potential. It comes down the axon. It hits the presynaptic area of the neuron, which is this area. When the action potential hits the presynaptic area, it causes calcium to be released in. The calcium allows the neurotransmitters to bind to the presynaptic membrane and then via exocytosis get excreted into the synaptic cleft, into the area between the synapse, between the presynaptic membrane and then the postsynaptic membrane. It diffuses across the synapse. It binds to receptors on the postsynaptic membrane, and they refer to it as a ligand. It opens gates on the postsynaptic membrane, and it lets sodium in. If it is excitatory, ES.
Can you make it bigger?
Yeah. If it's excitatory, it'll let sodium in. If it's inhibitory, it'll probably let chloride in or let potassium out. As you remember, your resting membrane potential is about minus 70 millivolts. Excuse me, potassium is positively charged. So if more potassium leaves, that resting potential is going to go down to minus 75, minus 80, and you've got to climb that much further until you can get up to the threshold for an action potential. Okay, they come across the synapse. They're referred to as a ligand. The opening of ligand-gated ion channels or chemically regulated ion channels. Graded potential. Now what happens is if one axon opens up and sends its neurotransmitter across, it may not be enough to cause an action potential on the other side. So the more neurotransmitters that fuse with the membrane and cross, the more there are, the more likely you're going to get an action potential in the postsynaptic membrane. It's got to be polarized enough that when it hits the axon hillock, that it will start an action potential. So, the channels open on the postsynaptic membrane. The potential changes depending on what ions channel. Sodium or potassium, it's going to be excitatory. ESPS, excitatory postsynaptic potential. If it opens the chloride or the potassium channels, it's going to be inhibitory. It's going to hyperpolarize. IPSP. ESPS, move the membrane potential closer to the threshold. Remember what we mean by threshold? Threshold is the membrane potential that kicks off an action potential. Generally about a minus 55 millivolts. So, the more positive potassium or sodium that moves into the cell, the more it brings it up closer to the membrane, to the threshold. If it's an ISPS, the membrane potential moves further from the threshold. Minus 75, minus 80, minus 90, somewhere down there. So it takes a stronger stimulation to get another action potential. Presynaptic, you have the axon terminals. The action potential is coming down the axon. It opens the voltage-gated channels, releases the excitatory neurotransmitter. On the postsynaptic, opens chemically or ligand-gated channels. You get an inward diffusion of sodium. You get the conduction of the excitatory postsynaptic potential. And at the axon hillock, if there's enough, it opens the sodium-gated channels, and the sodium goes in, and you have an action potential. This part is graded. Up to here, it's graded. When you start here from the axon hillock and down the axon, it's all or nothing. Okay?
Where is it? I'm sorry.
When you start, when it talks about a graded reaction, it may take a lot of these ligands to get across to get enough action potential down to the axon hillock to cause it to depolarize. But when it depolarizes from the axon hillock down to the next presynaptic neuron, or down to the motor neuron junction, it's a full potential. It's not graded, it's all or nothing.
It's the analogy of the bus with the 40.
Yeah, it's going to carry the 40 people whether there's 80 people there or not. Once that action potential starts, it's a full action potential. But until it gets to the axon hillock, it can be graded. You've got to get enough of these chemically operated gates open to let enough sodium in to get it to work.
And how is it called? All or nothing? Or
Yeah, all or nothing is fine. Okay, one of your neurotransmitters is acetylcholine. It opens when it binds to the channels, and it goes into the channels. It's excreted by exocytosis from the presynaptic membrane. It goes into the space, and then it binds to the receptors on the postsynaptic membrane. It's excitatory. In most cases, when you get into the brain and the autonomic nervous system, it can be inhibitory in some of the autonomic motor neurons. The autonomic motor neurons are motor neurons that you cannot control. Now, we're going to talk about a bunch of different receptors. And they're going to be called nicotinic, muscarinic, or other types of receptors. And they get their names by the fact that we found a drug that mimics them, or that can cause them to bind. So on a nicotinic receptor, it's an ACH, an acetylcholine receptor, but it'll also bind nicotine and do the same thing. So we call it a nicotinic receptor. They're found on the motor end plates of muscle cells. The motor, that's where the nerve attaches to the muscle cells and would cause the muscle cell to contract. They're in the autonomic ganglia and some parts of the CNS, and a lot of time we're getting into this. We'll get more of it in Chapter 8, and then as we get into the muscles and more into the CNS, you'll get it in more detail. Now, there's muscarinic. The second type of acetylcholine receptor is a muscarinic. That gets its name because of the muscarinic mushroom. Muscarine. It's a poisonous mushroom. They find that the toxin from that, probably enough people got poisoned by it, that they're looking at the toxin to see what's in there, and it binds to the postsynaptic membrane. So those receptors they call muscarinic. And again, it's in the CNS and the plasma membrane of smooth and cardiac muscles, and again in the autonomic motor neurons. Okay. Agonist is a drug that can stimulate a receptor. Nicotine is an agonist. Muscarine is an agonist. They're going to depolarize the postsynaptic membrane so that the threshold is reached easier and you get the next action potential. Now, atropine is an antagonist. Curare is an antagonist. What happens is they either attack the neurotransmitter and the synaptic cleft, or they bind to the receptors. So at this point, atropine binds to the muscarinic acetylcholine receptors, and the neurotransmitter can't work. Curare binds to the acetylcholine nicotine receptors. So the presynaptic membrane shoots all this nicotine type acetylcholine out, and it can't do anything because the receptors are already bound from the curare. Now, you're familiar with curare? Mm-hmm. South America, darts, it's from a tree sap. The Amazon Indians use it to paralyze. It's a pretty wicked poison. So they would tip their arrows with it to paralyze whatever animal and or other human they're shooting at. And what happens is you just go flaccid. And you have no muscle response to breathe or anything, so you suffocate. Atropine, I see atropine given if you have somebody with poison. that's got insecticide poisoning, something like that, because it reverses the insecticide poisoning, attacks the acetylcholine, and atropine will reverse that. Curare I've seen given in people with pancreatitis, and the muscles relax, they're put into a deep sleep for a week or 2 or 3 weeks to let the body relax enough for the pancreas to kind of heal itself. The stress of a pancreatitis can just feed itself, and until you get really, really sick. Okay, chemically regulated gates. Binding of the neurotransmitter to a receptor can open an ion channel. Now, we talked about this toward the end of chapter 7. Ligand-gated channels, or G-protein coupled channels. On a ligand-gated channel, when the acetylcholine or the ligand attaches, there might be 5 or 6 proteins that make up this channel. They just simply open up and let the sodium in. In a G-protein coupled channel, you have the ligand attaches, or the neurotransmitter attaches to the receptor. At the other end of the receptor, it has a G protein with an alpha, beta, and gamma molecules. One of those molecules breaks off, goes over, and opens the channel. Let's see if... okay, ligand is also an ion channel. Binding of the neurotransmitter opens the channel. Nicotine is a ligand-gated channel. Just like I said, it binds the channel opens up. Binding with acetylcholine opens a channel. It allows both sodium and potassium. Sodium in, potassium out. Because of the electrochemical gradient, there's more sodium on the outside because the sodium-potassium pumps have been pumping sodium out. And so sodium rushes in. Sodium comes in, potassium goes out. You get enough membrane depolarization to get an action potential. Now, you need enough of the neurotransmitter to come across, so you need a full action potential from the presynaptic membrane. Now you got that. Presynaptic is coming into the synapse, sending it down across the synapse and down the postsynaptic. So you have pre and post. Pre is before the synapse, post is after the synapse. You may need neurotransmitter for more than one presynaptic membranes into the postsynaptic. That's where your ESPS is. You get enough of the excitatory postsynaptic potentials, that kicks it up for the neuron, that kicks it up for the potential at the axon hillock to be enough for another total action potential, a depolarization. So here's your receptors. Here your acetylcholine binds, the receptors open, potassium goes out, more sodium goes in, your potential is raised up toward the threshold. Inward of sodium depolarizes cell, creating EPSP. They occur in dendrites and cell bodies. The binding of several molecules can be added together to produce a greater depolarization. This is your graded reaction. You need more molecules to move to start the action potential. This may reach the threshold for voltage-gated channels in the axon hillock and leading to an action potential. The action potential, again, is all or nothing. Here, it's graded because you're going to need several of these. It's not just one. You're going to need a bunch of them to get it to go. When it gets to here and starts down, if there's enough, it just goes. Period. So, right here, you start at a minus 70, you get, these are, could be molecules or tens of molecules of the acetylcholine binding and letting sodium in. So all of a sudden it's moving up until you hit a threshold of a minus 50. So you've got some sodium coming in, you've got some more sodium coming in, you've got more sodium coming in, and boom, you hit the threshold, you've got an action potential, and down it goes. Down it goes the X. This is why this is graded, because it takes more than one. And if you don't get enough to get up to here, it doesn't happen. It ends right there. And we'll be talking about that quite a bit in Chapter 8. Okay, comparison. This is your EPSP. Action potential, you depolarize. Acetylcholine or other excitatory neurotransmitters, they may or may not. The sodium channel opens, common channels for sodium and potassium open. So sodium comes in, potassium goes out. It's not a gated. So you have to have more sodium in, because for each positive sodium, you're losing some positive potassium. So it's not as efficient. Opening of potassium gates for repolarization, loss of intracellular positive charges. With time and distance, the more it fires, there's no repolarization in the postsynaptic membrane. So at some point, you can lose enough charges, but on the other hand, it can continue to fire. It's regenerated over the length of the axon. Your EPSP is localized. It's just from those dendrites into the axon. Positive feedback, yes. No. These, as long as you see a neurotransmitter coming across, it's going to work. It's going to keep going. Depolarization goes to a plus 30. Remember, you started at a minus 70 and went all the way up into a plus. This is not as efficient. It can get up to about 0 and that's it. But you need it at a minus 55 to get hit the threshold, minus 50. There's no summation. There's no summation, meaning when you have an action potential, it goes, period. Here you have summation because one ESPS probably won't produce an action potential. It takes a bunch of them. And sometimes you'll have 1,000 neurons coming in on the presynaptic and all coming down to one postsynaptic. A refractory period? Yes. No, you do not have a refractory period. They can continue to fire. They can continue to let sodium and potassium out as long as the neurotransmitters are there. Acetylcholine is affected by inhibition of tetrodotoxin, not by curare. ACH here is affected by curare. One's muscarinic, one is nicotinic. G protein. Remember muscarinic from mushroom? The nicotinic from nicotine. One is by curare, and that is muscarinic. Neurotransmitter. The receptor is separate from the protein that opens the ion gate. Binding the receptor opens ion channels by the G protein. Muscarinic receptors interact with the ion channels this way. You've got, let's see what it shows. Alpha, beta, and gamma.
Did you go back to the page just now?
This is in your book.
No, the one after it. This
Yeah.
one, thank you.
Muscarinic uses a G protein channel. Got it? 3 subunits, as I said, alpha, beta, and gamma. Binding of acetylcholine results in the dissociation of an alpha subunit. Either the alpha or the beta gamma subunit diffuses through the cytoplasm to the ion channel and opens it. Activation and deactivation of G proteins. Drug, botulism, inhibits the release of acetylcholine. Acetylcholine is inhibited. Again, there is no muscle contraction, it's flaccid. Again, you'll suffocate. Curare prevents the interaction of acetylcholine, again, with the nicotinic receptors. So either it doesn't release it or it blocks it. Botulism doesn't release. Curare, it blocks it. No, but you're going to have to know curare and strychnine and saxitonin, because it's in an example in your book. Okay?
Saxitoxin?
Saxitoxin, yes. Okay? But what you can see here is these drugs interfere with either the ion channels or the binding of the neurotransmitter or the release of the neurotransmitter. Any way it goes, you're in trouble. You have a little more time than you do if you've taken something like, like the cyanide, because you're dead in a couple of minutes with cyanide. This, I've seen people poisoned and still survive. Strychnine, they used to poison, all the wolves and that in Alaska, other places, coyotes, wolves. Use it as an animal poison. Okay. Acetylcholine opens potassium channels from some tissues or closes them. If it opens the potassium channels, muscarinic, if it opens the potassium channels, it's inhibitory. It's inhibitory because the potassium comes out, and what happens to your membrane potential? It doesn't say?
No, it doesn't say.
If the potassium is positively charged, your membrane potential is negative. If the potassium comes out, what's going to happen to the membrane potential? It's going to be more negative. It doesn't have the counteracting positive charges. That's why it's inhibitory. It moves the membrane potential down. If it closes, or closes potassium channels in others, where there's more potassium, then it moves the threshold up. If there's more potassium inside the cell, the threshold goes up. If there's less potassium, the membrane potential goes up. If there's less potassium, the membrane potential goes down. We're talking about potassium because it's the highest concentration in the cell. In the heart, potassium channels are open, creating what? ISPS, inhibitory postsynaptic potentials. It's hyperpolarization. It makes it harder for the action potential to go. It makes it harder for that nerve to fire. It slows the heartbeat down. In smooth muscle, the channels close, and it raises the membrane potential, and it's easier to depolarize, and then you get contraction of these muscles, and your stomach gurgles and moves and mixes, or your intestines move things down. This is autonomic. You can't control either of these. Okay? Acetylcholinesterase. the acetylcholine comes into the synaptic cleft and stays there, it's going to keep binding. You know, the ion gates and that, they don't have a regeneration time. They just keep open and let the sodium come in. So if that happens, the muscles are going to continually contract. You're going to have a tetanus, essentially. Nothing but is going to be able to relax. Hang on one second. Strychnine. It prevents ISPSs in the spinal cord, inhibits contraction of the antagonist. Okay, that's not quite what I was looking for. In the heart, acetylcholine. It hydrolyzes the cholinesterase. ACE is what? What does ACE signify? Enzyme. It hydrolyzes acetylcholine into acetylated choline. It goes back into the presynaptic cell, and it's reused. Acetylcholinesterase comes in, breaks up the choline. It stops the binding, and so you go into more of a resting phase. Okay, acetylcholine in the peripheral nervous system. Somatic motor neurons form interactions with the neuromuscular junctions. So the motor neurons are the ones that are coming down to the muscles to tell them to contract. And there you have the junctions. Neuromuscular junctions are essentially the synapse between the nerve and the muscle. As opposed to having a nerve-nerve synapse, dendrite to dendrite, axon to dendrite, axon to cell body, you have an axon to the muscle. The area of the muscle cell with receptors is the end plate. And so when it jumps there, it's an end plate potential. Got it? It's the same thing, except instead of going to another neuron, you're going right to the muscle. End plate, the open voltage gated channels result in an action potential, and the muscles contract. Certain drugs block it. Curare, again, antagonizes acetylcholine. It blocks the receptors, the muscles do not contract. It leads to paralysis, and your diaphragm is paralyzed, you can't breathe, you suffocate. When people have been put on Curare, they're always on a ventilator. They can't breathe on their own. Now another time they will use Curare is in electroshock therapy to relax the muscles. Because if the muscles are really tense, when they do that, the muscles will tear, you get a lot more damage. It's kind of like the drunk in the auto accident. It's so relaxed. They're relaxed. The person that they hit is all tensed up and generally suffers a lot more. Okay, Alzheimer's disease. Associated with the loss of the cholinergic neurons that synapse the areas of the brain responsible for memory. So, all of a sudden, you can't get these neurons across. You don't have the neurotransmitters to get across in Alzheimer's. And when we talk about memory in Chapter 8, you'll see that it's kind of a circular pattern. And if that stops, then the ability to know what's going on and to create memories goes away. Monoamines. They're regulatory molecules. Now let's just pause here for a second. Remember when we talked about homeostasis? and we talked about antagonistic reactions. Too long ago? That's chapter 1. I never thought we would go back that far. And you remember, let's talk about antagonistic reactions. You have, for instance, you have insulin that's lowering the blood sugar, and you have glycogen that's raising the blood sugar at the same time. You know, as the example in the book is, you've got, instead of just having the air conditioner on or the heater off, or the heater on and the air conditioning off, you've got them both on at the same time, and they're just very delicately balancing the temperature. This is what you're going to see in the CNS and the PNS all the time. You know, if you shoot all this acetylcholine in and wait for something to happen and then try to shut it off, you're going to get overreactions. If you've flexed a muscle, another muscle has to relax. So you have to have something that's going to the other muscle to tell it to relax. So if there's two reactions, these ESPSs and ISPSs are going on in conjunction all the time. Now, monoamines are a regulatory molecule from amino acids. Catecholamines, tyrosine, dopamine, derived from tyrosine, dopamine, epinephrine, norepinephrine. Serotonin, derived from tryptophan. You know, more tryptophan, more serotonin, more relaxed you are. What's full of tryptophan? Turkey. Who's ever awake after a turkey dinner? You just want to go to sleep. You got all that extra tryptophan. Though some people will argue that seems to always work for me. Histamine, from histidine, serotonin, dopamine, and norepinephrine are neurotransmitters. The monoamine action and inactivation. Like acetylcholine, monoamines are made in the presynaptic axon, released via exocytosis, which is the cell membrane, the vacuoles fusing with the cell membrane. The cell membrane opens up, out it comes. And released via exocytosis, goes across the synapse, bind to specific receptors. They're taken back into the presynaptic cell and degraded by monoamine oxidase. What does that? What's the big thing about pushing that back in? It's part of what the astrocytes do. So, here you have the monoamine is produced and stored in synaptic vesicles. It's come down, it's released as epinephrine, norepinephrine. It hits the receptors, and then it's picked back up and recycled. Again, as it comes down, the calcium gates open. The calcium gates open and allows these vesicles to attach to this presynaptic membrane. None of the receptors for these are direct channels. They always use what they call a secondary messenger. And most of it is cyclic adenosine monophosphate. It's most messengers for the catecholamines. It binds the receptor like a G, activates a G protein, sends the alpha subunit to an enzyme called adenylate cyclase, converts ATP to CAMP, and the CAMP activates the protein kinase. Any protein, any enzyme that ends in kinase phosphorylates. And it phosphorylates other proteins and it opens the channel. So here, norepinephrine to a receptor, it releases the G protein. It goes, takes the alpha subunit. It creates an adenylate cyclase. It creates AMP. The AMP goes over here and opens the channel. Just a couple more steps.
Can you do that, that process again, please?
Okay, it activates a G protein. Disassociates an alpha subunit. The alpha subunit binds to the enzyme adenylate cyclase. That converts ATP to AMP. AMP then activates another enzyme called the protein kinase, which and the kinase, as I said, always phosphorylates. It phosphorylates another protein, I'm not sure which one that is, and it opens the ion channel. Serotonin, using the neurons in the raphe nuclei in the middle part of the brain. It's implicated in mood, behavior, appetite, and cerebral circulation. Very calming until you get hit with LSD. And that will block the serotonin, and boy, if you've ever seen anybody on an LSD trip, it's something to behold. Specific uptake, it's reuptakes. There's reuptake, and it's the drugs that block the reuptake, leave the serotonin in there, are used as antidepressants. That's what Prozac, Palex, and Zoloft do. They block the serotonin reuptake. So the serotonin is still there to send the action potential, as opposed to something like acetylcholinesterase that stops it. This allows it to continue. There's a bunch of different serotonin receptors. Different drugs affect different ones. So you can take a drug for anxiety, appetite control, migraines. They just target a different serotonin receptor. Dopamine. It's in from the midbrain, from the nigrostriatal dopamine system, from the nigro area, the dark black area in the middle of the brain. And the mesolimbic dopamine system in emotional reward. You have two different systems with dopamine. One's muscular, motor control. One is in the mesolimbic system, and the mesolimbic system is an emotional system, circular system throughout that goes around in the brain. It works with emotional rewards, fear, things like that. In the nigrostriatal dopamine system, the neurons from the substantia nigra, they send dopaminic neurons to the corpus striatum. Important step in the control of an initiation of movements. Parkinson's disease is the degeneration of these neurons. You're not getting the dopamine across, you're not getting the transmission. You get this, one of them is, you go like this, pill roll. You walk into a room, you can see somebody doing this, sitting in the corner, kind of staring, doing like this. You can almost diagnose Parkinson's from across the room. You can see the pill roll. Parkinson's, it's by degeneration of the neurons. So what happens is we give them L-Dopa. Why don't we give them dopamine? Just give them dopamine. Doesn't cross the blood-brain barrier. L-Dopa will, and then be transformed into dopamine inside the CNS. But dopamine itself will not cross the blood-brain barrier. Now, well, we'll get into that in a minute, but if you give them too much L-Dopa, you're getting too much stimulation, you can push them toward schizophrenia. If you overdose a Parkinson's patient, you can push them toward schizophrenia. If you take a schizophrenic patient and put them on a dopamine blocker, you push them toward Parkinson's. So you got to kind of watch what's going on here. Mesolimbic dopamine system. Regions of the midbrain have dopaminergic neurons to the forebrain. It's involved in the emotional rewards. Schizophrenia is associated with too much dopamine. Drugs that treat schizophrenia are a dopamine antagonist, which you're actually pushing them back toward Parkinson's. Yeah?
Does it matter what type of schizophrenia it is?
What's that?
Does it matter what type of category of schizophrenia it is, or does it happen with all just schizophrenia?
I don't know. I just simply don't know. Norepinephrine, used in the central nervous system, peripheral nervous system. Sympathetic neurons in the PNS use norepinephrine on smooth muscles, cardiac muscles, and glands. Used by neurons in the CNS brain, it's associated with arousal, waking up. Amphetamines work by stimulating the norepinephrine pathways. Norepinephrine in the sympathetic, you're looking at your flight, and right. Other neurotransmitters, glutamate. We will see a lot of this in Chapter 8. The amino acid, it's used as a neurotransmitter. It's in the cerebral cortex, and it's excitatory. Energy required for ESPSs. You use an ATP. You're pumping sodium and pulling up, reabsorbing things. You use a lot of energy. 15% of your blood goes to the brain. And these we will just simply get in Chapter 8. There's 3 receptors for glutamate. And they work together in the memory storage in the limbic system. Limbic. And we'll get into that in Chapter 8. So we're getting to the end of 7, so just kind of, it's prepping you for Chapter 8. The neurotransmitter, amino acids, glycine, it is inhibitory. Binding of glycine opens chloride channels. Chloride is negative. What does it do to the membrane potential? Lowers it, moves it further away from the threshold, makes it harder to reach the threshold. Important in spinal cord for regulating muscle movement. Again, you have antagonistic muscle movement, and in the homeostasis, you have antagonistic balance. You have the air conditioning and the heat on at the same time. It's antagonistic. It's the same thing in the muscles. You're going to see this all through the CNS and the PNS. It's going to cause some muscle groups to relax because the nerves cannot reach the threshold. The action potential cannot reach the threshold because they're inhibited. So while the triceps contract, the biceps relax. It's got to work that way or else nothing gets done. Relaxing the diaphragm. Strychnine blocks the glycine receptors. So you can't relax. You stiffen up, you can't move your diaphragm, you suffocate. That's how strychnine works. GABA, it's used by 1/3 of the brain's neurons. It opens chloride channels. So is it inhibitory or excitatory? Inhibitory. Like it says up there, inhibitory. It's involved in motor control again. Degeneration of GABA secreting neurons in the cerebellum results in Huntington's disease. Huntington's disease is more correctly called Huntington's chorea, Huntington's dance.
Don't come in, don't do it.
What?
I was just joking. Don't do it, don't come in.
Oh, he's just locking us in. That's Huntington's. The GABA goes away. The nerves are not inhibited. They keep firing, and you get this chorea. You get this dance. The problem with Huntington's is it's not naturally selected against. It usually doesn't show up until the patient is in their mid to late 30s or 40s. By that time, they've generally already reproduced, they've bred, and that gene is dominant. So to get it, you got it. You pass it on, they got it. And you don't know until it's too late.
What does it do? What if your parents have it though?
You can do it.
Not necessarily. But yes. But then your parents get it at 40, 30, 40, whatever. You're 10 or 11 years old. The only way that you're going to combat it is by genetic counseling. You're going to have, somebody is going to have to say, I'm going to get my tubes tied, I'm going to get a vasectomy, something, so that I don't reproduce and pass that gene on. That is the only way that you're going to counteract this disease, unless we find some way of gene splicing to deal with it. And nobody bioethically is going to do that. And it's not a bioethics class. But this is the only way that, because nature does not select against Huntington's. It doesn't select. The only way nature would select against it is if this gene showed up before you reproduced. It didn't have a chance to pass it on. GADA, glycine, very similar. They're inhibitory. Neuropeptides, some chemicals, some peptides, and CCK, which we'll get into in Chapter 8. They're used as hormones or paracrine. What do we mean by paracrine? As opposed to endocrine, as opposed to exocrine. Paracrine. Within an organism, very close. It's right next. Whatever excretes it, the action is on cells that are right next to it, as opposed to an exocrine that might be going into the gut, or an endocrine that's going into the blood system. Paracrine is a hormone. It's secreted. It acts on the tissue right next to where it's secreted, or within a few cells of it. CCK, and we'll get into that. Substance P, again, we'll get into that in Chapter 8, mediates pain. Endogenous opioids. It's runner's high. You all heard the word endorphins. Endogenous opioids. Your body has a system for combating pain. It's got the endogenous opioids. They block neurotransmission and thus giving you pain relief. We do that with opium and morphine. We block it for you for extra. One physician who dealt with addicts said that if we could package an endorphin, it's probably 50 times more 50 times more powerful than heroin for creating dependence. It's that big. But obviously the body is able to control it. Endogenous opioids are polypeptides produced in the brain and the pituitary gland. The big one is, again, endorphin, you see, and enkephalin and some dynorphin. Nucleopeptides, why? It's in the brain. It's in stress response, circadian rhythms, cardiovascular control. We'll touch on that again. Endocannabinoids. Did you know your body made marijuana? Didn't make marijuana. It makes THC, the active substance in marijuana. Your body produces it naturally. That's the endocannabinoids. But it's very specific. They're short fatty acids. It's produced in the dendrites, and they're inhibitory again. They can be retrograde neurotransmitters, meaning that they can be released, by the postsynaptic membrane, go back to the presynaptic membrane, bind with the neurotransmitters there, and either cause an inhibition or excitatory. If it binds with a neurotransmitter that's transmitting GABA, which is inhibitory, and it stops that, your overall action potential is going to be excitatory. Or if it binds with an excitatory neurotransmitter, then it acts as an inhibition, it acts inhibitory there. It's released from the postsynaptic neuron. It's retrograde, so it goes back to the presynaptic neuron, and either blocks the secretion of the neurotransmitters into the synaptic space, or it helps. It helps reduce whatever else is blocking them. By, it's like, anybody that's the enemy of my enemy is my friend. That's kind of how it works. If the GABA is inhibiting the release of the neurotransmitter, the endocannabinoids block the GABA, then you got more neurotransmitter coming across, and you get your action potential. The enemy of my enemy. They produce IPSP producing, if they go back and they block, if they block the inhibitory protein in the presynaptic membrane, so that the excitatory producing neurotransmitters have a greater effect. It may enhance learning and memory, have certainly been shown to induce appetite. We give THC to people that we want to eat more. You can get it in a capsule. Marijuana, it's THC, but it's in the entire system. It's not controlled. And impairs learning and memory. Now, when we get into memory, we'll talk about the fact that in memory, you have to kind of slow down the neurotransmissions, especially into long-term memory, because you need time. Long-term memory is actually a physical thing, a growth of dendrites in the brain. Short-term memory isn't. That's just like water swirling around, and it swirls for a while and then it goes away. And if you ask me to remember a telephone number from here to walk into there, forget it, ain't going to happen. But long-term memory is actually a physical change in the brain. And unless you can relax and cut down the number of impulses that are coming across, it doesn't happen, or it doesn't happen as well. So we will get into that a lot in chapter 8. Nitric oxide. Do not mistake this with nitrous oxide. Nitrous oxide is laughing gas. Nitric oxide is a neurotransmitter. It diffuses. It doesn't need to have any vesicles. It just diffuses across the membrane and activates production of cGMP. And I'll have to get back to you on that. But anyway, in the PNS, the nitric oxide is secreted by the autonomic neurons, the digestive tract, the respiratory tract. It relaxes the blood vessels. It allows more blood. You've got a lot of food and you want to absorb the glucose. You want more blood into the intestines to absorb this glucose to get it to the liver. Well, another thing that it does is it relaxes the blood vessels in the penis. It allows the penis to engorge with blood and become hard or stiff, responsible for an erection. This is how Viagra works. It increases the nitric oxide. Now, the blood's got to come from someplace. Your body's only got so much. So you'll also see Viagra, i.e. nitric oxide, given into patients like with pulmonary hypertension. So it lowers the blood pressure, it lowers the blood in the pulmonary artery in the circulatory system, it opens it up, and it helps relieve the pulmonary hypertension. Carbon monoxide also does the same thing. We'll touch on it when we don't get into it. It's used in the olfactory epithelium and the cerebellum in the brain. And obviously you don't want too much carbon monoxide. And we'll get into that when we get into the respiratory system. ATP is a co-transmitter. It has to bind with something else. It's not a transmitter on its own. It has to bind. It's a purine. It binds to the purine receptors. It's released with norepinephrine to stimulate blood vessel constriction, and with acetylcholine to stimulate intestinal contraction. ATP. It's a co-transmitter, does not work by itself. Synaptic integration. Neural pathways. Divergence. Axons have collateral branches, so one presynaptic neuron can form synapses with several postsynaptic neurons. Branches out like a tree. It can convergence. You can have several different presynaptic neurons can synapse with one postsynaptic. That's convergence. You've got a bunch of neurons coming into one, or you got one spreading out to a bunch. Divergence versus convergence. Let me know when you're ready. Okay? Summation. Spatial summation due to convergence of signals into a single postsynaptic neuron. This is what we were talking about earlier. You've got all the excitatory and the inhibitory postsynaptic potentials. At some point, you got to do the math. They either add up to enough to get the membrane potential up to get an action potential, or if it doesn't happen, then it doesn't happen. You don't get the action potential. So here, ESP, excitatory. Here you might have a inhibitory until you finally get enough to get an action potential. But what happens is you've got to do the math. Somehow you've got to get from this minus 70 up to the minus 50. If you're secreting nitrous oxide, or if you're secreting a monoamine that's inhibiting, as opposed to the acetylcholine that's exciting, at some point you've got to get enough of one or the other up to get this action potential to go. Do the math. Synaptic plasticity. Use of a neuronal pathway may strengthen or weaken the synaptic transmission. Repeated stimulus enhances excitability. It's called long-term potentiation. This is used in memory, the formation of memory, especially in the hippocampus. And it's associated with the glutamate receptors. This long-term potentiation is very important in the creation of a long-term memory. Long-term depression occurs in glutamate-releasing presynaptic neurons stimulate the release of endocannabinoids. This suppresses the release of more neurotransmitters. In this one, it's inhibitory. What it does is it long-term depresses the firing of the axons. That's what long-term depression is. Synaptic inhibition is produced by the inhibitory neurotransmitters, glycine and GABA. They hyperpolarize the synaptic neuron. Which way does your membrane potential go with hyperpolarization? Down. Here, minus 70, Inhibitory postsynaptic potential, it starts dropping it down. Here you get an excitatory that's pushing it up. You get another inhibitory, it might move it down again. You get another excitatory, it might move it up. Do the math. Until you get up to this minus 55, you're not going to get an action potential. The body uses this to very finely control the action potentials. Presynaptic inhibition, endogenous opioids, and pain reduction. Neuron synapses on an axon of a second neuron, inhibiting the release of excitatory neurotransmitter. So you're doing it before you get to the synapse. Okay, that is the end of chapter 7. Tremendous amount of information, I understand. Why don't we take about 20 minutes and we'll start on chapter 8. And then about 9 o'clock, we'll, anybody that wants to stay for a review can stay, and anybody that thinks they've got it, come on. Yeah.
Okay, so just to double check, we have a test on Thursday or Tuesday?
You have a test on Thursday.
On chapters?
4, 5, and 6.
And the next chapter is, the next.
Well,
step.
7 is after 6.
7.
No, I'm just giving you that.
Oh, okay. I'm totally, I'm like so organized that this is like for me for a loop. So like, when's our next test after that?
The 12th of April.
So the 12th of April will be on chapters 7, 8, and then.
And if I get 9 and 10 done, it's going to be on 9 and 10.
Okay.
I can ask questions if you don't want to go.
But 8's a long chapter. 9's a short chapter.
It's not too bad. Like 36 pages.
What's that?
It's not too bad. It's about the same as the others. It's like 36 pages.
That's escape right there.
It's not working.